So I'm Ryan Cook. I'm a postdoctoral researcher here at Quadrum Institute in the lab of Evelyn Adriansens, and I see myself as a viral ecologist. So basically, I'm interested in what viral communities look like in nature. And when I say viral community, I talk about all of the viruses within a given environment, and the overwhelming majority of these will be bacteriophages, or viruses which infect bacteria, and that's what I'm most interested in. There's a lot of research into phage therapy, which is the use of bacteriophages as an alternative or addition to antibiotics. And one of the sort of central dogmas and most attractive things about bacteriophages is that they only interact with the bacteria. They are invisible to the human host. But in recent years, there's been some conflicting reports about that. So some great work from people like Jeremy Barr, Christina Dubrovska, Dwayne Roach and Paul Boyacki are showing that actually maybe bacteriophages do interact with the human host. Maybe they're internalized by human cells and digested, or maybe they're eliciting immune responses. This is something which we're looking at in my current work. So I'm taking sort of known bacteriophages and natural viral communities, mostly bacteriophages, and performing experiments with sort of organ-on-chip organoid systems to decipher any interactions that might be going on. For INPHARED, and I think most people's databases or bioinformatic tools, it usually has a selfish origin. So people will develop something which essentially is useful for them, and then maybe they'll share it with others. So during my PhD in the lab of Andy Millard at Leicester, although I was at Nottingham, we were doing a lot of bacteriophage genomics and viral metagenomics, where basically we're looking at bacteriophage genomes. And when you've sequenced either an individual bacteriophage and you have its genome, or you have a viral metagenome, so lots of bacteriophage genomes, one of your first questions is, are my genomes, or is my genome similar to those which have already been seen in nature? So basically, you want to provide genomic context for your sequences. Like, are there any close relatives which already exist, or is my sequence brand new? And so we were taking as many complete genomes from bacteriophages as we could, because we wanted that information ourselves so we could place our own data within that. And then it essentially grew over time into something which we then shared with others. In bioinformatics, there's a lot of interesting acronyms and names out there, some of which are probably better than others. For INPHRARED, which is Infrastructure for a Phage Reference Database, I think I was just brainstorming with myself, and I started with Phage Reference Database, and then I wanted to add an IN at the start to make it into a word, and that's where infrastructure came from. So there's quite a lot of filters which you can apply. The overall process is essentially that we take genomes from GenBank NCBI, but the issue is there's a lot of I don't want to say rubbish, but incomplete and maybe undesirable sequences on NCBI. So you can go to the RefSeq sequences, which are those which are very heavily curated, high quality, sort of exemplar sequences. But the issue with those is that that's a relatively small database and doesn't represent all of sequence phage diversity. The reason being just that it takes time for all of the QC and to have the exemplar isolates uploaded. So we want more information than that. So we look at all of the phage genomes on GenBank, but we know that a lot of these aren't great. So then we have to narrow that down. So we look for those with complete in the name. We try to remove any sequences where in the name there's something that suggests it might be an environmental or uncultured sequence. We only want those that have been isolated in the lab. We also look for certain tags in the GenBank record as well. So for example, if you had a sort of single phage genome from a single phage, you would upload it under the designation PHG. If you had a metagenomic sequence, you would upload that under ENV. So we filter out anything that's ENV, anything that's maybe a predicted prophage that hasn't been validated as being a sort of free-living bacteriophage. And that's pretty much it, really. So GenBank records are really useful in that there's loads of identifiers and metadata tags in there, and you can pull the data from that. Some submitters are more detailed than others, and some use the fields in a slightly different way to others as well. So yeah, one thing we're really interested in is the host. Which bacteria did this bacteriophage come from, or was it isolated against in the lab? So one thing you can do for that is pull the word which comes before phage or virus in the name. So Pseudomonas phage X. And then we think, okay, this was isolated against Pseudomonas. One of the issues of that, though, is that lots of people will actually put the sort of environment where the sample came from rather than the host. So rather than Pseudomonas phage, you'll have capybara phage. And we know that this virus doesn't infect capybara. It was probably just isolated from a capybara feces sample, for example. So for things like that, I do have to manually go in and clean them up. But for the most part, we can automatically pull, say, host data and other interesting metadata, sort of original analysis in the original paper. And the data set is very heavily biased. So the overwhelming majority of sequenced phages are tailed phages belonging to the class Cordoviricites, formerly the order Cordovirales, but that's gone now. But yes, so it's incredibly biased towards these specific phages. And that's largely because we're using the same sorts of samples and the same sorts of methods to isolate them. There's not a huge amount of variety there. And we're also using a fairly narrow range of hosts. And the reason for that generally is that these are important hosts which people are interested in. So for phage therapy, for example, people are interested in maybe Klebsiella pneumoniae, Pseudomonas aeruginosa, things like that. But by far and away, the host with the most phages isolated against it is actually Mycobacterium. And this is largely because of an incredibly successful program in the US called C-phages, where undergrad students are able to isolate phages, get them sequenced, and then deposit them. So we see a massive number of these Mycobacterium phages, many of which are very similar to one another because they're being isolated against the same host. It's a fantastic program, but it means that we have a lot of that particular type of phage. There are researchers looking to address this, though. So Michael Baim's lab, for example, has developed the phage disco method, which uses sort of isogenic strain pairs and fluorescence to try to find phages of a specific phenotype. And their method has massively expanded a group of phages called the Tectiviridae, for example. And I think that's what we need, basically novel, imaginative methods to try to isolate different phages rather than same samples, same phages, same hosts. So there's a couple of fantastic guides on this, two of which were in a special issue of the journal Phage, one of which came from Andy Millard's lab. I'm not involved with it. And one of which came from Evelyn Adriansen's lab. Again, I'm not involved with this either. One of them is how to name and classify your phage, and the other is how to annotate your phage. So the first bit of advice I would give is please do not name your phage P1, because there are hundreds of phages in the database named P1, isolated from completely different hosts and with no genetic relationship whatsoever. So firstly, make sure that your phage has a meaningful and hopefully unique name. That would be the starting point. And with regard to the submission, try to include as much metadata as possible. So location, maybe, the genome topology, whether it's actually complete or not, and ideally some high quality genome annotations as well. And again, these were born largely from selfish reasons. Basically, analyses I wanted to do myself, and then we made it available for others. And there are two which come to mind, really. So the first is that we output a MASH file, so users can use MASH to quickly compare their sequence against the database and see, are there any very similar phages in this database to my phage? And this has recently been kind of expanded upon by Andy Millard and Thomas Sikaritz-Pontine for their Tax My Phage tool, which works in a similar process, but then with some cool clustering. We also, at the time, were running vContact2 quite a lot, which is a tool for classifying phages based upon shared proteins, basically. So one of the outputs of INPHRARED is the two input files needed for vContact2, and a user can then combine their data with that data and run it through the vContact2 pipeline and see if their sequences cluster with any of those in our database. It was very much in the sort of One Health space. So we're thinking about animal health, human health, and the environment, which links them. And the main reason for the dairy farm being involved in that is because so in a typical UK dairy farm, there's a lot of cows and they produce a lot of feces. And this is generally all swept up, put into a large either tank or a lagoon, and it's stored as slurry, which we then spread in the environment as fertilizer. So we take this product, which has been in the cow gut, and we put it into our crop fields, and then potentially into the wider environment through leaching, but also into the sort of human food chain. So it's kind of important to know what's there if you're spreading something widely in the environment, right? So one of the things we really wanted to look at there was, are there any phages which could be making nasty bacteria or pathogens fitter and better able to cause disease? And we found quite a lot of virulence factors within the slurry tank for Streptococcus phages, which are one of the kind of leading causes of mastitis, which is a big problem in the dairy industry. So we were very much interested in phages in animal health from that side of things, but also then knowing what are we putting into the environment which could be affecting human health. There's a lot of conflicting reports. You'll read some studies which say that phages very widely carry antimicrobial resistance genes, and some which say they very rarely encode them. And I have to say that I'm in the latter camp. I think that antimicrobial resistance genes are generally over-predicted in phages, and that when scientists look to validate them experimentally, as I did during my PhD, you find that they're not actually antimicrobial resistance genes, which poses a very interesting question in of itself, because phages, they carry genes which make their hosts fitter, right? They carry toxins, fitness factors, metabolism genes. So why is it that they carry antimicrobial resistance genes so rarely? It's a bit of a confusing question. So obviously the bacterial community will shape the bacteriophage community, even just through simple things like host availability. If the host isn't there, then the phage won't be there. But equally, the phages which are there and infecting are putting selection and evolutionary pressures onto the host and directly shaping the bacterial community, as well as through temperate phages, which can integrate as prophages and directly influence the metabolism of their hosts. With regard to who changes first when there's an external factor, so let's say it's a person who's taken antibiotics, this is going to directly impact the bacterial flora and then maybe remove strains, so the phages which infect them are then no longer present because they have nothing to infect. But equally, if there are sort of sub-inhibitory levels of antibiotics which are stressing the bacteria, maybe then prophages are being induced from the bacteria, and so the phage is then shaping the bacterial community.